Dear colleagues, it is a great honor to be able to present the findings of my doctoral dissertation, Analysis of Evolutionary and Transmission Dynamics of Human Immunodeficiency Virus in Serbia and the Balkans. When it comes to HIV epidemic in the Balkans, there is a heterogeneous situation regarding the sizes of HIV epidemic in different countries, incidence rates. However, in the last couple of years, the dominant transmission mode is sexual, mainly through MSM contacts. In Serbia, first cases were registered in 1985, and by 2021, over 4,000 new cases were registered. And there are two hallmarks of HIV epidemic in Serbia. First, high male-to-female ratio, and second, MSM is a dominant transmission mode. It is now understood that the emergence of HIV in human population is a result of multiple host switching events, and of them the most important event would be the switching event of simian immunodeficiency virus between chimpanzees and humans, because the offspring of these strains are viruses grouped as a HIV-1, a monophyletic group M, which is a pandemic strain, and this strain is further subdivided into subtypes. When it comes to this subtype distribution in the Balkans, it is now understood that the Balkan is a melting pot of HIV diversity. However, subtype B is the most prevalent subtype in the majority of Balkan countries, with an exception of Romania, where subtype B is an emerging subtype. High replication and mutation rate make HIV a good model for phylogenetic analysis. Transmission clusters, defined by these analyses, can be a surrogate for epidemiological connections. However, there is a limitation regarding interpretation of direct epidemiological connections. Based on molecular clock algorithms, however, phylogenetic analysis allows studies of growth and decline in specific and key populations, as well as assessing the impact of migration on HIV spread. All of this makes phylogenetic analysis a useful tool in planning the targeted preventive strategies. Taking all of this into consideration, we aimed to investigate transmission dynamics and phylodynamic patterns of the HIV epidemic in Serbia generally and in subpopulation related to transmission mode and HIV subtype, to reconstruct the mathematical model of the current cause and future HIV incidence trends in Serbia, to explore the evolutionary dynamics and phylogeographic spread patterns of the HIV subtype B epidemic in the Balkans. We designed this study as a cross-sectional phylogenetic investigation of HIV and a long-term trend analysis of HIV epidemic growth in Serbia and 6 Balkan countries: Bulgaria, Croatia, Greece, Montenegro, Romania, and Slovenia. And when it comes to sequence dataset, we defined two distinct sequence datasets: a Serbian sequence dataset and Balkan sequence dataset. We use a plethora of different phylogenetic approaches and algorithms regarding the specific aims of our analysis. However, general criteria for cluster definition were as follows: monophily, similarity, statistical support, and reproducibility of evolutionary scenario. And based on these general criteria, 3 sets of different criteria were designed. Moreover, specific monophyletic clades from Serbian and Balkan sequence dataset were subjected to Bayesian molecular evolution analysis with an aim to estimate the time of the most recent common ancestor, to estimate the changes in effective population over time, and reproductive number over time. Moreover, for cross-border clusters found within the Balkan dataset, a phylogeographic analysis was done with an aim to analyze spatiotemporal patterns of HIV spread between different Balkan countries. Statistical analysis included logistic growth modeling of new HIV diagnosis in Serbia for key populations as well as general population, a latent class analysis of the studied Serbian dataset, and joint point regression analysis of HIV incidence trends in the Balkans for key populations as well as general populations. Initial statistical description of Serbian dataset revealed that Serbian dataset is a representative sample of Serbian HIV epidemic, illustrated with high male-to-female ratio and predominance of MSM as the most prevalent transmission route. Phylogenetic analysis of Serbian dataset revealed that nearly 1/3 of sequences clustered within the 19 transmission clusters, mainly connected to MSM and subtype B. However, 4 out of 19 clusters were connected with heterosexual transmission too. However, we also found 2 additional monophyletic clades with high nucleotide distance, over 4%, however, also high statistical support. When it comes to phylogenetic analysis of monophyletic clades associated with MSM transmission mode, we found that those clades were active during the whole study period, illustrated with reproductive number values above 1, and on the other hand, monophyletic clades associated with heterosexuals had a prolonged phases of stationary growth, illustrated with reproductive numbers below 1. When it comes to latent class analysis of Serbian dataset, we found 5 latent classes. However, in first latent class, we found young MSM, age less than 25, from Belgrade, diagnosed in the last 3 years of the study period, with 73% prevalence of other sexually transmitted infections, which represent, unfortunately, ongoing transmission trend. When it comes to logistic growth modeling of HIV epidemic in the general population and key populations, we found that HIV epidemic in Serbia is still in an exponential growth phase, mainly due to transmission within MSM key population. When it comes to phylogenetic analysis of Balkan dataset, we found that 68% of sequences clustered within 93 transmission clusters. Of them, only 4 were cross-border clusters. Distribution of sequences within these cross-border clusters were markedly different, from about 6% from Croatia and Romania, respectively, up until about 65% in Montenegro. About 40% of clustered sequences from Serbia and Slovenia could be found in cross-border clusters as well. When it comes to phylodynamic investigation of these cross-border clusters, we found that these are dated in the beginning and the mid-90s, with an exception of cluster 4 dated in the mid-2000s, and exclusively for cluster 4 is that the origin sequences were from Serbia and Montenegro only, whereas in other 3 we can call them ex-Yugoslavenian clusters, so to say. Phylogeographic investigation of the proposed cross-border spread revealed a reciprocal spread patterns between ex-Yugoslavenian countries with multiple introduction in Romania. When this spread is studied on the level of specific cross-border clusters, it is revealed that the origin of this cross-border spread is from Serbia or Slovenia. When it comes to joint point regression analysis of HIV incidence trends in different Balkan countries, we saw differing HIV incidence trends resonating with differing preventive policies implemented in specific Balkan countries. And to conclude, transmission dynamic of HIV spread in Serbia is linked to transmission clusters formation, which are connected to MSM transmission and subtype B. Phylodynamic analysis of active clusters coupled to LCA analysis confirmed young MSM with concomitant STI as the main target group for planning structured preventive policies. Presented results imply that HIV epidemic in Serbia is still in the exponential growth phase, in particularly related to the MSM transmission. HIV subtype B dispersal pattern within the Balkans is mainly driven by local transmission clusters, with cross-border clusters observed mainly between countries of former Yugoslavia and Romania. Regional spread of HIV within the ex-Yugoslav space has continued after countries' breakup, whereas the spread of subtype B through multiple introductions to Romania resonates with the changing pattern of travel and migrations in Europe. HIV epidemic remains an important public health problem in the Balkans, in spite of the recent downward trend in the number of new HIV diagnoses seen in some countries. Thank you for your attention.